Good day, students. I am your student teacher, John Paul Biburila, and I am here to discuss about our new topic, which is entitled Glycolysis Process. But before we start, let us have our learning objectives. At the end of the lesson, students should be able to, first, define glycolysis and name the enzymes involved in each steps of glycolysis. Second, appreciate the importance of the process of glycolysis in energy production. And lastly, explain the steps of glycolysis accurately and concisely using appropriate terminology. So now let us begin to our proper discussion. So now let us define first what is glycolysis. So it states here that glycolysis is a series of reactions that extract energy from glucose by splitting it into two 3-carbon molecules called pyruvate. So it means that the glucose have 6 carbon molecules and it splits up into two, or it breaks down into two, and the end product is called pyruvate. So next, glycolysis comes from a merger of two Greek words, glyx, which means sweet, and lysis, breakdown or splitting. Also, glycolysis is an ancient metabolic pathway, meaning that it evolved long ago, and it is found in the great majority of organisms alive today, such as human beings. Also, it is stated here that it is a universal pathway given by Emden, Meyerhoff, and Parnas, and this is often known as EMP pathway or MP pathway. So I have a question here. So where does glycolysis occur? Very good. Glycolysis occur in our cytoplasm of our cell. So next, glycolysis is an anaerobic process. So what does it mean? So it means that glycolysis does not require oxygen to proceed. So next, each reaction in glycolysis is catalyzed by its own enzyme. So what does it mean? So it means that in every steps or stages in glycolysis, there are enzymes responsible for it. And glycolysis have 10 stages or steps. So therefore, there are 10 enzymes responsible for each steps. The most important enzyme for regulation of glycolysis is called phosphofructokinase. So what is his contribution to the glycolysis process? So it catalyzed formation of the unstable compound or molecule. So siya ang nag-catalyze ng formation ng mga unstable na mga compound or mga molecule. So let us deepen our understanding about phosphofructokinase. So it states here that phosphofructokinase speeds up or slows down glycolysis in response to the energy needs of the cells. So what does it mean? So it means that if the phosphofructokinase is present in the process of glycolysis, it speeds up the process. While if the phosphofructokinase is absent in the process of glycolysis, it slows down the process. So now, let us view together the figure of our glycolysis process. So here is our glycolysis process. So as you can observe or see, there are more structure on it, right? And there are 10 stages or steps in the glycolysis. But we're not just focusing on the structure. So let's begin in the step 1 of glycolysis. So step 1, phosphorylation. A phosphate group is transferred from ATP to glucose, making glucose 6-phosphate and ATP. So it means that ang phosphate group na galing kay ATP ay nag-transferred kay glucose. And it makes into glucose 6-phosphate and ADP. Alam naman natin na ang glucose ay mayroong 6 na carbon molecules, di ba? So, kaya siya naging 6-phosphate kasi ang phosphate group na galing kay ATP ay doon na-attach sa carbon 6 ni glucose. So next, glucose 6-phosphate is more reactive than glucose itself. So, si glucose 6-phosphate ay mas aktibo. And the addition of the phosphate also traps glucose inside the cell that can't cross the membrane. So it means that mag-add na naman tayo ng phosphate group tapos si glucose ay mananatili sa cell at hindi makakalabas ng basta-basta sa ating membrane. Therefore, our responsible enzyme for the step 1 of glycolysis is hexokinase with the cofactor of magnesium 2 plus. And here is the figure of the step 1 of glycolysis. And as you can observe, glucose turned into glucose 6-phosphate. And also, the figure shows that the phosphate group is attached in the carbon 6 of the glucose. So next, step 2, isomerization. Glucose 6-phosphate is converted into its isomer and will become fructose 6-phosphate. So it means that si glucose 6-phosphate ay nag-convert into fructose 6-phosphate. And actually, fructose is still a sugar that composed of 6 carbon molecule, the same as glucose. And the difference between glucose and fructose is their structure. And our responsible enzyme for the step 2 of glycolysis is phosphoglucose isomerase. And why phosphoglucose? Because we will respond to the glucose 6-phosphate. And also, the figure shows that glucose 6-phosphate turned into fructose 6-phosphate. So next, step 3, phosphorylation. A phosphate group is transferred from ATP to fructose 6-phosphate, producing fructose 1,6-bisphosphate. So what does it mean? So it means that ang phosphate group na galing kay ATP ay nag-transfer na naman kay fructose 6-phosphate. So mayroon na tayong dalawang phosphate group na utang galing kay ATP. So ngayon, si fructose 6-phosphate ay naging fructose 1,6-bisphosphate na kasi mayroong dalawang phosphate group na na-attach sa ating structure. So now, our responsible enzyme for the step 3 of glycolysis is phosphofructokinase with the cofactor of magnesium 2 plus. So, which can be regulated to speed up or slow down the glycolysis pathway or the process, as I said earlier. Now, the figure shows that fructose 6-phosphate turned into fructose 1,6-bisphosphate. And there are two phosphate group attached now in our structure. So next, step 4, breakdown. Fructose 1,6-bisphosphate will break down into two 3-carbon molecules. So it means that si fructose 1,6-bisphosphate ay magdi-divide na naman into 2. So we have glycerol aldehyde 3-phosphate or G3P and dihydroacetone phosphate or DAP. And our responsible enzyme for the step 4 of the glycolysis is fructose bisphosphate aldolase. And this is the figure of the step 4 of glycolysis. And as you can observe, si fructose 1,6-bisphosphate is divided into 2. So we have DAP and G3P. So next, step 5, isomerization. Dihydroxyacetone phosphate or DAP is converted into glycerol aldehyde 3-phosphate or G3P. So it means that if we had to produce DAP, it will be converted into G3P. So therefore, we are going to produce 2 molecule of G3P. And our responsible enzyme for the step 5 of glycolysis is phosphate isomerase because DAP and G3P is isomer to each other. So next, step 6, phosphorylation. Glycerol aldehyde 3-phosphate or G3P, one of the 3 carbon sugars formed in the initial phase, loses 2 electrons and 2 protons, reducing NAD plus to NAD H and producing H plus. So it means that ang isa sa carbon ni G3P ay magre-release or maglulus ng dalawang electrons at saka dalawang protons. So, ngayon, si NAD plus ay nakaatang na kukunin niya ang mga nai-release na mga electrons at protons. Tapos kukunin niya ito at magproproduce siya ng hydrogen. So, this reaction releases energy which is used to attach another phosphate to the sugar, forming 1,3-bisphosphoglycerate dehydrogenase. So, it means that in the surrounding of NAD plus, there are another phosphate that are going to attach in the sugar or in the structure. So the responsible enzyme of the step 6 of glycolysis is glycerol aldehyde phosphate dehydrogenase. So as you can observe in the figure, we have G3P releasing electrons and we also have NAD+, and in his surrounding, mayroon tayong phosphate group. So mag-a-attach ito sa ating panibagong sugar or structure. So si G3P will become into 1,3-bisphosphoglycerate. So next, step 7, substrate level. 1,3-bisphosphoglycerate donates one of its phosphate groups to ADP, making a molecule of ATP and turning into 3-phosphoglycerate in the process. So it means that si 1,3-bisphosphoglycerate ay magdodonate ng isang phosphate group kay ADP. So ngayon, we only have one phosphate group in the structure. So, our responsible enzyme in the step 7 of glycolysis is phosphoglycerate kinase. As you can observe in the figure, we have two phosphate group that attach in the structure. So, galing kay 1,3-bisphosphate glycerate. So, it will turn into 3-phosphoglycerate. Next, step 8, isomerization. 3-phosphoglycerate is converted into its isomer and it will become into 2-phosphoglycerate. So, it means that instead na naka-attach si phosphate group sa carbon 3, doon na siya maa-attach sa carbon 2. So, kaya naging 2-phosphoglycerate siya. So, the responsible enzyme of the step 8 of the glycolysis is phosphoglycerate mutase. So, as you can observe in the figure, we have 3-phosphoglycerate turned into 2-phosphoglycerate. So, next, step 9, dehydration. 2-phosphoglycerate loses a molecule of water, becoming phosphoenol pyruvate or PEP. So, it means that we are going to release or loses a water. So let's deepen our understanding about phosphoenol pyruvate. So it states here that it is an unstable molecule poised to lose its phosphate group in the final step of glycolysis. So it means madaling matanggal ang ating phosphate group sa end product ng ating glycolysis process kasi nga unstable ang kanyang mga molecule. So our responsible enzyme in the step 9 of glycolysis is enolase with the cofactor of magnesium 2+. So as you can observe in the figure, we have 2-phosphoglycerate turned into phosphoenol pyruvate. So we have the losses of water or H2O. So next, step 10 or the final stage of the glycolysis process, substrate level. PEP readily donates its phosphate group to ADP, making a second molecule of ATP. So 2 ATP produced. So it means that nabayaran ang ating dalawang utang na phosphate group na galing kay ATP. So next, as it loses its phosphate, PEP is converted into pyruvates or pyruvic acid, the end product of glycolysis. So it means that after losing phosphate, it will become or it will converted PEP into pyruvate or pyruvic acid. As I said earlier, na madali siyang matanggal. So our responsible enzyme in step 10 of glycolysis is pyruvate kinase with the cofactor of magnesium 2+. So as you can observe in the figure, we have PEP or phosphoenol pyruvate turned into pyruvate. At as you can observe, wala na siyang naka-attach na phosphate group sa ating structure. And that is the process of the glycolysis. As for your assessment, just click the link below that I provided and submit it by tomorrow. Thank you and God bless us all.